Okay, students, we are now going to conclude chapter number 5 by dealing with the last two categories of our macromolecules. In the last lecture, we looked at carbohydrates and lipids, and now we want to move on to proteins and nucleic acid. Now, of the four macromolecules, I would say that by far the most diverse and the most complex are going to be our proteins. And so we're going to start with them first and deal with these very unique and very diversified type of molecule, and then we will move from that point on to our nucleic acids. Two things to keep in mind when it comes to dealing with our proteins. Polypeptide is a polymer of amino acids connected to a specific sequence, and so we're going to talk about that. We're going to deal with polypeptide chains, and these polypeptide chains then can eventually lead to what we call a protein, and then a protein's function is going to depend upon its specific conformation. And what we mean by conformation is its specific three-dimensional shape. And so when you move on into anatomy, one of the things that we talk about in there is a term that is known as the complementarity of form and function. And what that basically means is that the form of an organ, the form of a tissue, the structure of a cell is going to be directly related to its function. It is designed to do what it's supposed to do. It is shaped in such a way that it can carry out that particular function. Same is true on a molecular level. So when we talk about our proteins, we are dealing with molecules that have specific shapes, specific conformations that allow them to do specific things. So first, we need to talk about what proteins are. They're very instrumental in about everything the organism does. Proteins are important for structure, storage, transport, signaling, movement, defense mechanisms. When we deal with the human body, antibodies are formed by proteins. So we've got a huge variety of things that our proteins are going to be doing. We have tens of thousands even inside of the human body, each one having its own unique structure and each one having its own unique function. When it comes to our proteins, we are talking about a polymer that is constructed from a set of 20 monomers. These are called amino acids, and when you put these amino acids together, you end up having peptide bonds, and as the peptide bond continues to lengthen, we have what are called polypeptides. So if we were to say the monomer was the amino acids build into polypeptides, polypeptides then build into our proteins. And again, they are going to have a specific conformation, which is then going to lead to its specified structure and also its specified function. So, excuse me, amino acids consist of 4 components. So we want to talk about each of those, and here are the ones that they have. There's actually 5 components if you count this central carbon. So you've got a carbon that is structured in the middle, and then from that carbon we're going to find these following components that we find. So first we have a central carbon. Second, we're going to find a carboxyl group, a COOH group. This is going to be the acidic component of our amino acid. We're also going to find an NH2 component, or what is called an amine or amino component, and that amino is going to give off some basic characteristics. There is a hydrogen atom that is bonded to this, and then following the hydrogen atom is what we call an R group. The R group, or the variable group, is actually going to be the component of the amino acid that gives it a different function from the others that are in this particular class. 19 of the 20 are going to have the same basic base. What we mean by that is they're going to have the central carbon, the amino, the carboxyl, and the hydrogen atom, and then they're going to have a different R group, and that R group is going to be the difference maker. You're also going, the physical and chemical characteristics of the R group determine the unique characteristics of that amino acid. So that's going to be the key component to our amino acid in determining what our function of the amino acid is going to be. One group of amino acids has a hydrophobic R. These are going to be our nonpolars, and you can see them there. We will talk about these in class, so don't get too concerned about them at this particular moment. But you can see here we find 9 such nonpolars. And then we have our polars. You can see 1, 2, 3, 4. You can see 6 of them here. That would be having polar groups, making them hydrophilic. And then we also have those that ionize. We have some that are acidic and basic. And this is because of their general makeup. And so when they are put in when they are put in water, they will ionize, and as they ionize, you're going to see them have some cellular pH, whether it's acidic, meaning that the pH is going to drop, or they're basic, which means that the pH is going to go up. When you put them together, you end up having what is called a peptide bond. Now, very similar to our glycosidic linkage, in which we take a hydroxyl group and we take a hydrogen atom and put them together, and when you pull the water out, you end up having this glycosidic linkage, and you build a disaccharide from 2 monosaccharides. The same is true when it comes to a peptide bond. In a peptide bond, you are going to take an OH from the carboxyl group and an H from the amine group, and when you put those together, you are going to form water. So again, we have a synthesis reaction going on here, dehydration, pulling water out. And when that happens, you have formed what is called a peptide bond, and the peptide bond is going to be between the carbon and and then the second amino acid is going to be the nitrogen. So you form a bond between the carbon and the hydrogen, or the carbon and the nitrogen rather, and that forms what we call a peptide bond. So again, very similar to our glycosidic linkage are these peptide bonds. As you repeat this process, you are going to form a very lengthy polypeptide chain, and as this polypeptide chain continues to get larger and larger and larger, we're going to end up having our functional proteins, moving into our protein region. And so polypeptides are going to start, once we start adding the amino acids together, they can range from a few monomers up into the thousands of monomers. The function is going to depend on its specific conformation. Again, here what we're talking about is shape is going to determine function. Okay, so that's the thing I want you to basically get from this. Shape determines function. So as we look at this, you can see that this particular protein has a particular shape, and whatever shape we have is going to determine what the function is, depending upon how it's twisted, folded, and coiled into a unique shape. A protein's specific conformation determines its function. In almost every case, the function depends on its ability to recognize and bind to some other molecule. So if we're dealing with enzymes or we're dealing with antibodies, a couple of these examples, or even neurotransmitters, they're going to be what we call molecule specific. There are going to be certain things that they bond to because of the shape or the conformation that they have. In our proteins, this is why they're so diverse. This is one of the reasons why they are possibly the most complex of all of our macromolecules, is the series of levels of structure that they have. If we go back to carbohydrates, pretty simple. You take carbohydrates, you put them together, you form a linkage, that linkage forms a disaccharide, and on and on and on. When we get to our proteins, we have a variety of levels of structure. There are 3 levels that are going to be undergone by all polypeptides, and then we can also move into a 4th level. So we want to take a little bit of time to go through this. I'll try to do the best I can to explain this in as simple a fashion as I can. So let's begin. First of all, you have primary, secondary, and tertiary structures. In what I want to maybe say is regular language, this would be 1st level, 2nd level, and 3rd level structures that are going to be used in single polypeptides. When we move to very highly complex polypeptides to form individual proteins, we have what is called quaternary structure, or a fourth level of structure. So let's look at these. Primary structure. This is very simple. The primary structure of a protein is simply the sequence of the amino acids. What order are the amino acids actually in? Kind of like a train. Okay? If we have, let's say we have 5 cars. You've got car A, B, C, D, and E. Okay? Now we can put them together in that order, A, B, C, D, E, or we can actually change that. Maybe we want the order to be A, B, E, C, D. Maybe we want the order to be A, C, E, B, D. In an amino acid or in a polypeptide chain, if we change the order of those amino acids, then that is going to change the polypeptide that we are forming. Okay? So I'm hoping that that's kind of getting sinking in. Primary structure is simply going to be in what order are these amino acids actually placed in. Now here is an example in which we actually have the order substituting something in that should not be there. Okay, this is what we call sickle cell anemia or sickle cell disease. This is caused by abnormal hemoglobin. Hemoglobin is the oxygen-carrying protein in your red blood cells. Red blood cells' primary function is to carry oxygen throughout your system. And if your red blood cells are not developed correctly, then this is going to cause the oxygen-carrying capacity to drop, and the less oxygen that reaches your cells, the less cellular respiration you can undergo. Therefore, it's going to cause issues with the human body. So if you can read this, and also in your notes as you go through, you're going to see that you have a sequence of amino acids. And notice that in the second one that there is a replacement. In the place of number 6, glutamine, we actually have valine that has been substituted. And when that happens, you go from a normal shaped red blood cell into red blood cells that have these sickle shapes. And a sickle shaped cell is not going to have near the oxygen carrying capacity. Number 2, so number 1, first level or primary level of structure is going to be the amino acid sequence. Number 2, we have the secondary structure or the secondary level of protein structure. And this is going to be based upon its folding. How do the bonds actually interact and cause this particular protein to fold? And so we have 2 different types of folding. We have what are called alpha helices, or an alpha helix shape, and what are called pleated sheets, beta pleated sheets. And this is all going to be depending on the hydrogen bonds that are occurring. Some are going to actually have hydrogen bonding that occurs in such a way that you get a helical shape, kind of like a spiral staircase. In other classes, you're going to end up having these beta pleated sheets. And so it's a different type of folding. So if level 1 is amino acid sequence, the second level, or the tertiary level, the secondary level of structure is going to be how are the hydrogen bonds interacting to cause the backbone? Is the backbone folded in a helical shape, or is it folded in a pleated shape? Tertiary level of structure, this would be level 3. This is going to be determined by the interactions among the R groups and between the R groups and the polypeptide backbone. Okay, so again, amino acid sequence is going to determine the primary level, the first level. Second level is going to be determined by how is the hydrogen bonding occurring to form a backbone. Do I have a helix or do I have a pleated sheet? Thirdly, we're going to have interactions among the R groups as well as the R groups and that backbone. So as they begin to interact with one another, we start to move into a third dimension. Okay? We start to move into a 3D structure, and this 3D structure, or what we call this level of structure in which we are three-dimensionally shaped, we start to see a variety of functions take place. Sometimes we're going to have fibers, sometimes we're going to have globular proteins, and that all is going to depend upon how are these bonds, how are these atoms interacting one with the other. Here we have what is called a disulfide bridge. This is, of course, going to exist between the sulfhydryls, and because of this, because of these disulfide bridges, you actually see an interaction in which we are pulling a part of this shape together. We're holding it together, and so the interaction allows this protein to have a very unique shape. And because of its unique shape, because of its unique structure, we then end up having something that is very unique and has its own basic function. The fourth level, if we get to this level, now not every protein is going to reach this level, but sometimes we get to this level. And what we find here, the quaternary level of structure, is going to be when we take two or more polypeptide subunits and we put them together. What we're saying here is, by themselves, they're not going to be proteins. But when you put them together, they make a very complex protein. Two examples are collagen and hemoglobin. So if you can see here, you've got these three fibers that are intertwining, three polypeptide chains intertwining to form collagen. In this diagram, we see that you've actually got four that are coming together, and these guys are forming what we call hemoglobin, that iron-containing protein, iron-containing pigment that is found in our red blood cells, which is what gives blood its very unique reddish tint because of the iron that is contained in there. So we've got iron contained, we've got heme and iron, and then these chains, these four chains come together, and we have hemoglobin because of that interaction. So as we look at this, we have the amino acid sequence. Here's your primary level. Okay, how does it fold? Does it fold as a pleated sheet? Does it fold as an alpha helix? How are they interacting with one another, and what kind of folding do we have? Then we move into tertiary. What are the interactions between the individual R groups, and what are the interactions between the R groups and the backbone? Gives it some three-dimensional structure. And then we move into the quaternary level, and in that quaternary level we've got these multiple polypeptide chains coming together to form one unique functional protein. Okay? Now we do need to talk about the shape and the function in regards to homeostatic balance. The physical and chemical conditions will determine what happens to a particular protein. So for example, if we alter the pH, this can cause the protein to denature. Salt concentrations, temperatures, and a variety of other factors can cause an unwinding or a breaking in the hydrogen bond, which will alter the shape. And if you alter the shape of a protein, you've altered its function. It is now no longer able to do what it was designed to do. And so we have to be real careful, and our bodies are designed in such a way so that it is going to resist these pH changes or these salt concentration changes, temperature changes, trying to keep everything in balance so that our proteins remain at an optimum. In spite of the knowledge of three-dimensional shapes of over 10,000 proteins, it is still difficult to predict the conformation of a protein simply from its primary structure. Okay, because there's a whole lot of things that can take shape, that can take place as you move to secondary, tertiary, and on into quaternary levels of structure. Moving through the last 3 slides rather quickly, just read over that, and if you have any questions, we'll talk about it, some things that I'm not going to be too concerned about on your test. Lastly, we want to talk about our nucleic acids. This is our 4th class. We've talked about carbs, we've talked about lipids, we've dealt with proteins, and now we want to deal with the last of our macromolecules, which are our nucleic acids. In nucleic acids, we're going to be dealing with the storage and transmission of hereditary information. We want to look at the monomers, which are actually called nucleotides. We'll talk about what a nucleotide looks like and what happens when they come together. Inheritance is going to be based upon DNA, and then protein synthesis, we're going to deal with another type, which is called RNA. Amino acid sequence of a polypeptide is programmed by a gene. A gene is simply a particular segment of DNA. DNA is comprised of individual it is a particular nucleic acid, just like RNA is, and this particular segment, whatever the length of it is going to carry with it a specified gene for a specified trait, which is then going to allow us to inherit individual things from our parents. So first, let's deal with the 2 types that we have. There is RNA and there is DNA. RNA stands for ribonucleic acid. DNA stands for deoxyribonucleic acid. The primary difference between these two, DNA is going to be responsible for your heredity, for your inheritance, and it's also going to send the message through RNA for protein synthesis. So DNA is the one that is the controller, but RNA is the one that carries out those particular mechanisms, carries out those particular sets of instructions. When we are moving this information from DNA and beginning to code for specific proteins, we have to get the information from inside the cell to outside the cell, or rather the nucleus, inside the nucleus to outside the nucleus. And this is done when DNA actually synthesizes a molecule of RNA that is known as messenger RNA. And this messenger RNA is then going to move out of the nucleus into the cytoplasm, and then we're going to begin the process of protein synthesis, the building of these proteins. So if you kind of follow this flow chart, you've got genetic information comes from DNA. DNA is transcribed into mRNA. That mRNA then takes those instructions out into the cytoplasm, where a protein is going to be built because of the presence of ribosomes. So if you follow this chart, synthesis of mRNA inside, it goes out of the nucleus, moves to a ribosome, and because of those ribosomes, individual amino acids are being brought in, linked together through peptide bonding, and when this happens, we develop and build our individual proteins, which can then be used throughout the body. Nucleic acids come from monomers called nucleotides. As you add these nucleotides together, you get a polynucleotide, and these polynucleotides are eventually going to form our 2 classes of nucleic acids. Each nucleotide has 3 parts. You have a nitrogen base, and there are 5 different nitrogen bases that we're going to look at. A pentose sugar, pentose, 5 carbon sugar, and a phosphate group. When you put those 3 things together, you have what is called a nucleotide. So here is an example. Here's our nitrogenous base. Nitrogenous base connected to a particular pentose. There are 2 pentoses, deoxyribose and ribose. And then we have the phosphate group. This is going to form the backbone of our nucleic acids. Our different bases, we have what are called pyrimidines and what are called purines. The major difference here are your pyrimidines are 6 carbon, is a 6-carbon ring, and your purines are actually going to be double rings. And you have cytosine, thymine, and uracil. You have adenine and guanine. Those are our 5 bases, and then our 2 ribose, or our 2 sugars, our 2 pentoses, deoxyribose, DNA, ribose, and RNA. Okay? The nitrogen bases are going to, again, come in these two varieties, pyrimidines and purines, and each of them are going to actually they're going to be unique to the individual sugars. And we'll talk about that here in just a minute. Hopefully that's not too confusing, but we'll talk about that here in just a moment. So we got our polynucleotides is going to be our repeating backbone, and these backbones actually will lead to our two different types of nucleic acids. There are four. I want to close this out really by looking at the differences between DNA and RNA. I know there's still a few slides that we've got here, but I want you to understand the major differences between these two particular nucleic acids. First of all, there's going to be a difference between DNA and RNA when it comes to their shape. Okay, the shape is going to determine the function, just like we find in proteins. But the difference in structure is going to be RNA is a single polynucleotide chain. So you've got a one-sided chain. DNA, on the other hand, is going to consist of two polynucleotide strands that actually are going to wind around each other, around an axis, an imaginary axis, to form what we call a double helix. Now this was first proposed in 1953 by Watson and Crick, who eventually would go on to win a Nobel Prize for their development of the structure of DNA. So here we have the backbone of our DNA. You can see the structure. You've got this double helix, and again your RNA is going to be a single straight polynucleotide chain. So one, we've got a difference as far as the structure. Double helix DNA, single strand RNA. The second difference is the sugar. What is the sugar contributing to the individual nucleic acids? And what we find there is in DNA, we have deoxyribose, that's its pentose sugar. And then we have ribose, which is the pentose sugar for RNA. So deoxyribose versus ribose. So there's a second difference. The third difference are the bases that we find in these two different molecules. DNA is going to have adenine, cytosine, guanine, and thymine, A, C, G, and T. RNA is going to have adenine, cytosine, guanine, and uracil. So there is one replacement. Thymine in DNA, uracil in RNA. So there are the two basic, there's three basic differences there. Now if we want to go to a fourth difference, we would say that DNA is the genetic material. It's also the material that controls what takes place, and RNA is going to be protein synthesis. It's because RNA comes out of the nucleus, and then we are able to build our proteins from that. There's what we call complementary base pairing. In these base pairing rules, what we find is that a particular purine will bond with a particular pyrimidine, and they will only bond in these in this manner. They will not bond in any other way. So adenine is always going to partner with thymine. Guanine will always partner with cytosine. You will never find them partnering in any other way. Adenine is never going to partner with cytosine. Thymine is never going to partner with guanine. It's A to T, T to A, G to C, and C to G. So everywhere you would find a T on one side of the DNA, the other side is going to be an A, because they are going to complement one another, and that's why we call them complementary base pairing. And if we know one side of our DNA, we're able to figure out what the other strand of DNA is, because they're going to be held together with these hydrogen bonding based on this complementary base pairing. Now we can use DNA and proteins, as far as our class is concerned, as tape measures of evolution. Now what this means is that the more similar the DNA molecules are, the more similar the species will be in the concept of evolution. So if our DNA sequences are if there's great variation in our DNA sequences, then there's going to be great variation between the individual species. However, if there is little difference, then we're going to see a closer relationship. So basically what we're saying here is, and from an evolutionary standpoint, the more similar the DNA sequencing, the more related the organisms happen to be. The more variation there is, the less relationship there is going to exist between those two particular species. They're going to be more distantly related, the more variation that we find in there. So for example, here's a sequence of amino acids to form a particular chain, looking at hemoglobin of different organisms as compared to the hemoglobin found inside of humans. So if we compare these to humans, notice that gorillas, there's one difference, and only one difference in that amino acid chain. When we look at a gibbon, there are 2. Rhesus monkey, there are 8, and then the mouse and the frog are 27 and 67 respectively. So what this would say then is that the gorilla and the gibbon and the rhesus monkey in that particular order are going to be closer related to humans than the mouse and the frog happen to be. So over these last 2 lectures, what we have looked at is the concept of macromolecules. We've talked about monomers becoming polymers through dehydration synthesis, and we've talked about polymers becoming monomers through hydrolysis. So we're either going to be building something or be breaking something apart. We've talked about carbohydrates. Carbohydrates come in 3 varieties. We have the monosaccharides, disaccharides, and polysaccharides. We've talked about our lipids, which are primarily hydrophobic. We've got triglycerides. We've got our phospholipids, which have a hydrophobic and hydrophilic portion. And we've also talked about our steroids, in which we find testosterone, estradiol, and also cholesterol, which is very, very important. We've dealt with proteins. The primary thing to remember about proteins, 20 amino acids come together to build those. And we have to deal with 4 levels of structure. We've got amino acid sequence, which is primary. The particular backbone, is it a helix? Is it a pleated sheet? That is going to be secondary. Tertiary is the particular 3-dimensional conformation. What is the relationship between the backbone and the R groups and the R groups themselves? Then there's quaternary, when we actually have an association of multiple 2 or more polypeptide chains coming together to form one particular protein. And then lastly, we have our nucleic acids. The backbone there are going to be nucleotides. Nucleotides are formed by a ribose sugar being connected to one of 5 different bases that contain hydrogen, and also a phosphate group. We've got DNA, which is hereditary material. We have RNA, which is responsible for our protein synthesis. Write down your questions so that we can address these on Monday and Tuesday. I hope that I have covered everything in as much depth as I can regarding this particular chapter. And if you have any questions, be sure to ask me on Monday and also on Tuesday.